Welcome to another broadcast from mindyourhealthcast.com. I'm sure you were waiting anxiously for my next subject to drop on you, and I believe that you will actually find it very intriguing. Back along the way, of course, we have on numerous occasions talked a bit about DNA. However, today, DNA is going to be the major subject. And it is called DNA from Mechanic to Ghost. Now, that may sound strange, and incidentally, part one. That may sound strange, but you'll see what I mean when we get down the road into this up to our neck. It will begin to make some sense, I hope. And it might even to me. I have been around to watch the development of the discoveries related to DNA, the early assumptions, the infighting between researchers who were wanting to be the first to actually demonstrate the structure of the DNA molecule. And so there was a period of time, in the 40s especially, when there was a lot of hustle bustle going on about what is the actual structure of DNA. Now, the whole idea came about many years before that, that there was something that passes on information from one generation to the next. But it was not very well known. In fact, it was hardly known at all. Other than the fact it happened, there was not too much information about how this could possibly happen or what caused it to happen. And so it became apparent from some of the very early work of some of the... Now remember, a lot of these guys came about at a time when certain amounts of knowledge related to the human body and to the history of humankind were more limited. And so based on what they knew at the time, you would say that they were ahead of their time in many respects, but also, in my mind, they were, let's say, much more prone to jump to conclusions that later down the road ended up not being quite accurate. But because they were pioneers and because they did step out, I think they're to be applauded, because they were willing to gather what information was available and organize it in such a way that we could propose some kind of theory about what's going on. Because I remember up into this early 1900s, it's when I would say a lot of significant things started happening in the world of physics, in the world of chemistry, in the world of biology. And so they were working pretty much in the dark, I'd have to say, up until that time. And even then, they were only seeing pieces here and there. And if you can imagine having no map and trying to assemble a map, but to do it only from seeing pieces of information as opposed to the whole puzzle laid out, even though it might be disassembled if all the pieces are there, at some point you're eventually going to assemble them the proper way. But when you're just looking at a piece here and a piece there, and from that somehow you're trying to create a map or a picture, it can become very difficult. Now, you remember before we talked about the fact that the map is not the territory. This is a statement that's used frequently in NLP. And a map is a drawing or a representation that we create that supposedly represents something. And the more information we have, the more accurate it can be. And so we do that to ourselves. We have created a map of reality, and Therefore, we act as though that is true. And if we're open enough and new information comes along, then we're willing to alter that in such a way that it becomes more in conformity with the information we have. Now, as you're going to see, and especially if you go study it on your own, which you really should, you're going to find that there's been a lot of assumptions and presumptions related to DNA that as time has gone on, has changed dramatically. In many respects, it's actually become the opposite of what was originally thought. And that will become more clear as we wade through the quagmire of the concept of DNA and genetics and review some of the things that along the way transpired because of the way we saw things. So I have a very detailed history or timeline of everything related to the genes and genetics going way back to 1859. It encompasses 13 pages, and so we're not going to wade through all this 13 pages, but only to show you that there's quite a history of steps along the way that led to where we are. Now, I have another one called Genetics, the Early Days, which is more workable because it basically just hits the very main turning points along the way that led us to where we are and where it appears we could be going. Now, there's all kinds of issues related to genetics and what they can code for. And so we're going to look at some of those issues, and before we're finished with this whole subject, we're going to be introducing you to some of the newest information from around the world that is probably going to not only grab your attention, but it's going to maybe provoke you enough to either look into it or say, oh, well, it's all hogwash, forget that. We know what the real story is, and we're sticking to it. So, what is DNA and how did it all come about? Well, some of the early work that led to the whole concept was the work of a researcher by the name of Charles Darwin, and the writing of a book that came about from a lot of his travels and studying the different species of animals and so forth. He came up with a concept called the theory of evolution, based on what's called natural selection, which in essence means that you have a gene. Well, they didn't call it genes. They just knew that you inherited something that made you unique as a person, and that things that your ancestry had seemingly were passed on to you, in particular in your family, in general, and mankind. So, based on that, he said, in its simplest form, there's some type of information that's transferred from one generation to the next that it determines something about mankind as a whole and in a particular family. And so the fact that it changes on occasion led him to believe that occasionally that information got disturbed or interfered with and hence mutated. Now, remember, he didn't know anything about all this DNA and so forth at that time. This is 1859. But he knew, and pretty much anybody who paid attention knew, that you would see certain traits in your kids that your grandparents had or that you had, and so forth. So, I mean, there was a general consensus. There was some connection from your ancestry to where you are. And again, not just of you, but with you and mankind as a whole. In other words, humankind. But he said sometimes something interferes with this occurrence, and that interference is a mutation, and therefore it changes the person who receives that mutation slightly from their ancestry, and maybe from mankind ancestry. And so with that, that means then that you may be better adapted at living in the changing times that you were born into, or you may be less adapted. So if you are not capable of coping with the environment, you may expire. And if you do, then that mutation that you had expires with you. Unless you had children before you expired, which could happen, of course. And or that mutation may make you better adapted to the new environment, and therefore the survival capability is greater, and whatever that trait was then gets passed on to your ancestry ahead of you. I shouldn't use the word ancestor, to your progeny ahead of you, and you change. In other words, how the people were behind you in your ancestry weren't so capable, had they been here now, to survive this change. And so he said it's a very long, slow process that man gradually adapts to their environment by mistake. In other words, it's a random event. There was nothing planned, program about it. Just suddenly something interfered and made a change that made you more capable of surviving. So that's the old original concept or theory of evolution. It was a mistake, but it was actually a useful mistake. And people who had mistakes that weren't useful didn't survive, and therefore that passed away and was gone. And it kind of made some sense to a lot of people, except for people who were in the various church groups and various religious groups, period, who said, this is ridiculous. Man was created perfect to begin with, and that's the way he has been, is, and always will be. And hence this whole thing is contrary to God, the Bible, etc. So this big factional fight came along. However, around the same time as Charles Darwin, there was another researcher from France whose name was Dr. Lamarck, and from his observation over his life, he determined that whatever it is that we pass on from one generation to another is not fixed but mutable. That means that if you are exposed to some kind of poison and your body ends up altering itself in a way that it can handle and deal with the poison, then you have actually changed your chemistry or changed your coping capability. And if then you do that, it alters whatever this information is that you're passing on to the next generation, so that the next generation now is capable of utilizing the change which occurred in you, and henceforth, down your chain of progeny, you're going to be passing it on. So, everybody in the scientific community, I should say most people, I guess maybe not everybody, looked at both of these concepts, and for whatever reason, they liked Darwin's better. It seemed to fit some of the other thinking that was going on, such as Newton's idea that the universe is clockwork, and it just keeps going a certain way unless something were to interfere with one of the gears and make a change, it would continue to go that way, or if someone put a different gear in and altered the whole mechanism. And so everybody thought, again, I say everybody, I'm generalizing, the bulk of the scientific community felt that Darwin's idea was more in line with what they had been thinking, and so henceforth, Lamarck kind of fell into the background. And boy, did he fall into the background. I mean, it's just like Antoine Béchamp, as we mentioned earlier, versus Pasteur. Once Pasteur was adapted as the correct idea out of that period of time, Béchamp totally disappeared. I mean, you never hear from him. I went through school, I studied biology and science and chemistry and you name it, everything to do with the body and health and sickness, and never did the name Béchamp come up. The same thing when I studied electronics. There was a big controversy between Tesla and Edison. And boy, all of a sudden, Tesla just disappeared. It was Edison. Edison everything. And so the same thing is true with the DNA, or what became later known as the DNA, when it was between Darwin and Lamarck. Lamarckism, as it was called, just was pushed out to the side. Nobody ever bothered to talk about it again. After all, you know, it was not right anyhow, because we all knew that what Darwin had introduced was the correct answer. And so, as time went on, by the time this was 1859, by around 1865, there was a priest by the name of Gregor Mendel, who started studying something about plants and culturing plants. And out of that, he did some experiments and was able to demonstrate that there were certain traits that were carried that could get passed on, but sometimes they were in the background, but they didn't get expressed in a generation, meaning it could jump over generations. And so he was looking at color of bloom and pink and white bloom. He could take a pink and a white plant and mix them, and all the next generation may all be pink, but then on the next generation, white could pop out. And so he laid out some basic rules of what he called traits, how these traits were passed on. And bit by bit, it awakened people to the fact that there's some things were happening that bore further looking into. Okay, then the next significant, now there were some other steps along the way. As I say, if you look at the longer timeline, it's pages and pages long. We don't want to have to go into every little step, but the next significant thing that popped up was in 1910. Thomas Hunt Morgan, he understood or saw that there was something called genes on chromosomes, and that took it a step further. So now we know that there's something there that's carrying this information, bottom line. Now at that point, of course, it wasn't called DNA because they hadn't quite gotten to that point yet. So that was a big step, because he observed this, and bit by bit it started to get into where they did some experiments with fruit flies and so forth, and carried it on. Then in 1944, now again there were several things that happened in between, but it would be of no benefit. And if you want the complete history, you can go look it up. But there's no big benefit other than to say the next significant thing that got involved was in 1944. There was a researcher by the name of Barbara McClintock, and she discovered something called transposon, or posons. Transposons were genes that got relocated. In other words, whatever was going on, they kind of thought up to this point that the genes were fixed, but now she demonstrates that they can change location, or were given the common term jumping genes. So that just added a bit of flavoring to the stew pot, so to speak. And so then the next place that we really get to significant, and this is when I kind of got in the picture. You know, I was in high school at the time of Watson and Crick, James Watson and Francis Crick, which are the two big individuals that are regarded as the grandfathers of the genetic structure. Now, up until this point, bit by bit, different chemists, they found out there were nucleic acids, they found out there were chromosomes, they found out that there were things called genes on these chromosomes, but they didn't have any idea how this all fit together. What was the actual structure of it? And so one of them was from the U.S., the other was from Great Britain, and they both at Cambridge. And they were doing all this study. In fact, if I recall, Watson was the one from the U.S., and that he was there on an advanced degree program. And the two of them became friends, and they became very enamored with this whole idea of the genetic code and the DNA, which is deoxyribonucleic acid, is what the full term is. And so it was too much of a mouthful to spit out every time you talked about it, so they just abbreviated it DNA, and now everyone knows it as DNA, and there's hardly anybody that knows much of anything that hasn't heard of DNA, and pretty well know that somehow it's involved with the cells of the body and what you inherit from your ancestry, etc., etc. So that's pretty well, I would say, general, although I could be mistaken. Some of the younger generation now, when they get interviewed, don't seem to know a whole lot about anything, so I accept what's the latest style for Hollywood, etc. So I have to be careful when I generalize like that. It may not be true. But at any rate, these two gentlemen, who were very good friends, were in Cambridge, and there was a lot of work being done to determine the structure of DNA. And the race was on as to who is the scientist that's going to crack the code of the structure of DNA. It was almost a given that whoever did it was going to receive the Nobel Prize. And of course, that's the holy grail of science and research, is to be a recipient of the Nobel Prize. That indicates that on a worldwide basis, you're acknowledged as having made some giant stride forward to the furtherance of whatever area the award is given in. It could be literature, it could be medicine, it could be chemistry, and in other words, there's a series of categories. But they knew that in biology, they were going to get it, whoever came up with this. Now, one of the big contenders happened to be, in this country, we had Linus Pauling, who was considered the master of chemical bonding. He had written the textbook on the way atoms join together to form molecules, which is called chemical bonding. And he recognized, by then, of course, quantum physics had been coming along, and he understood the quantum concepts of the hydrogen bonds, and in fact, all kind of bonding. And so, as I say, in fact, he had received, I think by then, 2 Nobel Prizes, at least one. And so he was looking forward to receiving the Nobel Prize again for cracking the code of the DNA. So here's Watson and Crick, 2 students at Cambridge, who are playing around with this whole thing. And there's another lady there by the name of Rosalind Franklin, who was heavily involved in trying to come up with that structure. Now, what they had found at that point was that if you combine two atoms together, and you wonder what the actual way that they come together, how they bond. The only way you could find out for sure was to do something called X-ray diffraction, crystallography. In other words, you take the substance and you crystallize it, then you show X-rays through it, and they scatter off of the parts inside the molecule and form a pattern on an X-ray film, and from studying that pattern you can determine where the various portions of these atoms are located to form a molecule. So she was doing studies with the X-ray crystallography, and the Watson and Crick, they were looking at her work, and they were looking at some of the other studies that were coming along. They knew the fact that the chemical constituents that make up the DNA by this time, and so it was a matter of how do we bring this all together and be able to demonstrate the form. So while they're pondering all of this, now I'm throwing in some of the politics and other stuff in the background so that it becomes a little bit more interesting. While this is going on, here is Linus Pauling over in western United States. I believe it was in Washington or Oregon, where his institute was set up. They were doing all this study, and he was sure, being the expert, worldwide recognized expert on chemical bonding, he was going to figure it out. Now, it finally had kind of gotten down to how is the backbone of this molecule arranged. Now, anybody who has seen a representation of DNA today, of course they all know. But back then it wasn't known. Was it a single backbone that carried the code? Was it a double background? Or was it a triple background? They just weren't sure. They knew the chemical constituents, but how did they actually join together to form this helix? And so while they're pondering all this, one night all of a sudden somebody comes running up and says, Pauling has done it. He just published a paper in Science, which is the worldwide science journal. And if you want to receive recognition for or against you, that's the journal to publish in, because they will only publish your material if they peer review it. And so Pauling comes along and he proposes a triple helix. And right away, they had already tried that. They actually made models and tried to get it to fit together into a triple helix, and it wouldn't work. And the reason it wouldn't work was because it violated some of the principles of chemical bonding as had been discovered and published by Linus Pauling. In other words, he violated some of his own findings in coming up with the triple helix model. And as soon as they heard that, they said, he's going to be very embarrassed because it's in violation of his own textbook on chemical bonding. We know it couldn't be a triple helix. We already considered that. So they continued to work on their model, and they made actually pieces and had this big table, and they were working, trying to come out with ways to get them to fit, like a jigsaw puzzle. And finally, after looking at the X-ray crystallographs and all of the things that they had brought together, one night it all came together by putting the right amino or nucleic acids together in the right sequence with the backbone, which is a sugar phosphorus backbone, because it's deoxyribose, ribose sugar, the backbone. It all fit together. And they thought, wow. And they immediately sat down and wrote a very short article illustrating how our DNA helix comes together to form the DNA molecule. And wow, as you might guess, the world was astounded and said, well, of course, we should have known that, as always happens after the fact. But at any rate, they did in fact receive the Nobel Prize And so the DNA became the holy grail of the secret of life, the secret of nature, the secret of passing on information. I mean, it was considered to be one of the major discoveries. So they then started to work with understanding more about it. And very rapidly, as happens in the world of science, there are those people who take over pretty rapidly and give it an official tap on the shoulder. They dub you on the shoulder with the sword and pronounce, this is it, and that's it. And so that would become the standard position on DNA. Now, there's a lot more we can go into it, but we want to talk for just a little bit about the standard. The standard is actually called the central dogma of DNA, and a little bit about the structure of DNA. As I'm sure you've seen, it's a double helix. Now, it's not a spiral. A spiral is something that starts small and gets bigger and bigger in one plane. A helix stays the same diameter, but just keeps going higher and higher and higher. Like everybody talks about a spiral staircase, in most cases it's not spiral, it's helical. It's spiral only is if it gets larger as you go up, but that's not usually the way a staircase would go. So there's a double helix, and that double helix has become like a symbol. As soon as you see it, you think, aha, DNA, so everybody knows that. So the standard central dogma was, okay, now we know that there's a double helix structure, and we know that this double helix structure has cross links that are made up of something called nucleic acids. Okay, so we also know that there's two ways that the DNA chain is communicated or translated. One is that it is recopied when cells replicate. In other words, when a cell is weakening and wearing out, and the body's going to replace the actual code inside, and we won't even go into that process at this point, other than to say splits the genetic into two groups, and the new cell then gets half with a new half added on to match it. The old one keeps the old half and adds a new half to it until it dies off. And so that's called replication. So usually, barring again some kind of influence, you get an accurate replication in the new cell of what was in the old cell. If there were bad genes in there, as we sometimes refer to them, they would be bad genes in the new cell. So replication was one way. The second way that the DNA was carried, or did something, let's say, in the central dogma was that it transcribed RNA. We add a new factor here. It transcribed RNA with the information of a portion of the DNA. I have a picture here. I don't know if you can see it or not, and we'll take you through this little picture sequence. First of all, here is the DNA and the double helix, and then on the other side are the nucleic acids that make it up. Now, I don't know if you can see that or not, but you can see the DNA, and you can see the two groups of nucleic acids. Now, they are guanine and cytosine, which are the two that join together, and this is what Watson and Crick determined. And there's adenine and thymine. Now, always in combining, those pairs are the only ones that will combine with each other. Guanine and cytosine will always join together, and adenine and thymine will always join together. And so that's what holds the two helixal backbones together, is the cross-link between these nucleic acids. So the nucleic acids attach to the backbone on one side and into the right nucleic acid molecule, which is then attached to the other side. And so those form like steps or rungs in a helical ladder all the way up as the full length of the DNA strand. Okay, so now we mentioned RNA. And so RNA, again, I don't know if you can see this or not, but I'm holding them up in the case you can. Here's the DNA, here's the RNA. RNA is just one strand, a half a strand. Now, they're both made from the same thing, so we have RNA ribonucleic acid, and we have DNA deoxyribonucleic acid. And so hence the letters RNA and DNA. Now, RNA is basically like half of a DNA strand, except instead of using thymine to match to the adenine, it uses uracil and adenine together. Now, don't ask me why, and I'm not sure if anybody knows why, but for whatever reason, and I suppose in the wisdom of the body and things that do and can happen, it becomes important to substitute uracil on the RNA. Now, that RNA also is sometimes referred to as messenger RNA because it's carrying information from a portion of the DNA which the body has said we need this code to synthesize some amino acids into a protein because we need that protein to carry on because the old is worn out. In other words, part of the rebuilding process. So I have a little illustration of the two strands of DNA opening up and RNA being built off of one half of the strand. And so it's like it's building the complement of that. And from that, the way these sequences come out is that we have letters like an alphabet from the DNA that actually code for the amino acids. Now, as I say, the DNA separates because it gets a message we need a particular protein, so that portion of a strand of DNA opens up and splits apart and allows a duplicate to be made of one of the strands, which is RNA and referred to as messenger RNA. Then that leaves the nucleus of the cell out into the cytoplasm and is picked up by a special structure in the cytoplasm called a ribosome. And the ribosome utilizes the information on the messenger RNA like a template to start building the protein. It also has stop and start codes in it so that it just doesn't keep filling the cell full of the synthesized protein. So there is inside the cell another type of RNA called transfer RNA that picks up the right amino acids that are called for as the ribosome is building a chain of protein and puts in the right amino acids in the right sequence to create a protein molecule, which then enfolds on itself according to certain rules and forms a three-dimensional protein molecule. Quite a fascinating process. Okay, so the process of transcription is that the RNA is a transcription of the DNA portion containing the information, and once it gets out and is picked up by the ribosome translates the information on the messenger RNA and adds the amino acids together into a chain called protein. Sounds pretty mechanical, as we mentioned, and pretty cut and dried. Now, this is kind of the central dogma of DNA. Pretty clear cut. Makes a lot of sense, I guess. No. we're going to add the variations. This is the basic picture. Okay? The basic picture is that here we have this code in the cells of the body that is like a library of information that contains everything that you need to repair and rebuild the body. Also, remember that hormones contain chains of amino acids, hence a protein factor, and also that enzymes are chains of amino acids. So when we're transcribing from the genetic code a piece of information, we are in a sense building new enzymes as they're needed, new hormones as they're needed, and new protein to build new muscle or whatever we need to build in our system that requires protein molecules. Which means that at any given time the cytoplasm of the cell has to contain all the amino acids that are necessary, and that's called the amino acid pool, both in the cell and in the blood continuously. That's why we eat proteins and break it down through our digestive tract to get amino acids for the amino acid pool, so that it's available for us to rebuild the body, make new hormones, make new enzymes to keep everything working. And now talk about a meticulously designed being. I don't even call it a machine, because it's way beyond a machine, because of the complexity of the processes that are going on, and going on simultaneously, and going on without us consciously having to even think about it. Now this, to me, in itself is miraculous. But this is only the beginning, because as we get into the next one or two broadcasts, it may take 2 broadcasts to get through it. But for a long time, this was the fixed idea about the genetic. Genetic is fixed, and as soon as we solve this, we right away, we're going to say, wow, everything's fixed. Our behavior's fixed, our brain is fixed, our body is fixed, everything's fixed by the genes. If we get bad genes from our ancestry, then we're going to be a bad person. And so out of that grew something called eugenics. I don't know if you've heard of it or not. It was big in the early part of the century, or even before they knew all about the structure of the DNA. They knew there was something being passed on, some information, and that they thought it was fixed. And they maintained that even after the structure of the DNA, although it kind of was pushed back because it was unpopular. But eugenics means getting rid of bad genetic carriers. In other words, if you come from a family my father was a criminal or a murderer, then I'm going to be a criminal or murderer because it's in my genes. So therefore, the best thing to do for me is, number one, don't allow me to marry and produce children, because then they too are going to be murderers, and we don't want any more murders around. So the best thing to do is either sterilize me or dispose of me in some way, so that that bad gene can't be passed on. And that was called genetics, or eugenics. Now, you think that sounds absurd or outlandish? Hitler operated on the basis of eugenics. In this country, it became very popular in the 30s, to the point that Harvard University had a college degree in eugenics. A lot of the research was being involved, was involved in the whole concept of eugenics. If you don't believe me, go back and read it. And as a consequence of all this, now I want you to give this some thought. Talk about witches' hunts. As a consequence of this, thousands and thousands of people were forcibly sterilized so they could not pass on this bad genetic information. Thousands of people were forcibly, not voluntarily, but forcibly. And in Hitler's case, he decided, now I know it's not popular to talk about, but the history is the history. Are you going to hide it over that this is what happened? Hitler thought, just eradicate the whole race, and then you'll be done with all that whole genetic line that can't contaminate our pure Aryan German genetic line. Now, I want you to think how the implications are of information sometimes, whether true or false, what can happen to people in a society based on that. With that, I guess we are going to have to call this to an end today. But I'm going to read you about something real fast called Creating Life as We Don't Know It. Scientists are changing the rules for how DNA works. So, now I've given you the simplistic process. I've given you the standard central dogma. And it got to the point, I'm sure you heard, oh, there's a fat gene, there's a gene for this, there's a schizophrenic gene, blah, blah, blah. Okay, so the next big step was the Genome Project. And that's what we're going to talk about the next time is the Genome Project first, before we get into all this, because that was to determine the genetic structure, the whole code for a human being. And therefore, with all this knowledge, we're going to be able to go in and cut out certain genes and splice in new ones, and we're going to solve all the ills of mankind. Okay, our time is up, so until the next time, I hope I've whetted your interest enough to really get you into some of the mind-bending ideas coming up next. Until our next broadcast, be sure and go to our website, mindyourhealthcast.com. And it was nice being with you again, and until the next time, be healthy.